Greetings, everyone, and welcome to the Pan Future Society podcast. Today is Saturday, March 25th, 2017. I am your host, Sean, as usual. Something you'll hear me reference a little later in the show is that science is a tool. Like any tool, it could be used for good or bad. And what I mean by that, to describe by way of metaphor-ish description, a hammer can be used to put a roof on a house, or it can be used to bash someone's head in. So science, like any tool, it's a thing. Some people say science is a verb. Science is a way of understanding the world, of exploring the world, of figuring out how the world works. And really, everything is science. I'm badly paraphrasing something I read a while ago, that, beyond being born and breathing and eating and reproducing and defecating and then dying, everything else is science. Harnessing fire is science. Making tools is science. Everything that we've done as humans to make our lives easier or better, figuring out how to build a tent, build the wheel, farming to better produce food, crossbreeding animals to make better livestock. All of that is science. Whether or not people doing it at the time, 10,000 years ago or 5,000 years ago or 1,000 years ago, really thought of it as science, they were using science, the tool of science, to do this. You need to have an ability to remember what you already did. In a lot of ways, science is also history. You do something and take a look at what the results were, and if they weren't what you expected, you make some changes to your process, and that is also science. So don't be afraid of science. It can be used for bad. Yeah, that is true. But science itself isn't that way. It's the people. It's the people that we have to talk to. And as a society, as a culture, as a whole, we decide what are the acceptable ways to use science. So the topic for the main segment today of CRISPR-Cas9 and gene editing, it's fascinating, amazing science, and it's also a hot-button issue for ethical considerations that people will be talking about, and you need to face it. And the best way to face difficult questions is to have as much information as possible. I don't see how anyone can make good decisions without knowing anything about what they're talking about. So hopefully the show here will help you out with that. Now on with the news. Some nanomaterial news today from the Science magazine. Science magazine is something I subscribe to as a member of the AAAS. But you can also, I will link to an abstract of this so you can kind of see the summary that I'm going to give you and get a few more of the details if you do not subscribe to the magazine. The Hall-Petch relationship is a term describing a metal's increasing hardness with decreasing grain size. So the smaller a grain of metal material is, the harder it gets. However, when those grains are really, really small, nano-sized, that relationship breaks down and they become soft. A team of researchers working with a nickel molybdenum alloy altered the molybdenum composition and, as it says here, annealed the samples at just the right temperature which stabilize the grain boundaries in their nanograin samples. So the short version is nanomaterials made of metals, when they get to a certain size, get soft, which may not work very well, but now a new technique for making them could provide a way of making super hard coating for certain nano machines or whatever you happen to be working with. And now, it came from outer space. In space news this week, the United States Air Force's robotic X-37B spaceplane is now the longest-running mission in that program's history. As of March 25th, the current mission of the X-37B has been orbiting the Earth for 675 days. This beats the previous mission by 1 day, and we actually don't know when this one is coming back down, so it could continue on for quite some time. The Air Force, of course, has classified everything about this, and basically they say it will land when it's done doing whatever it's doing. So this is the fourth mission for this mystery space plane, and it has been orbiting the Earth for quite a while. Despite the length of this mission and it being a record for this particular program, it is not the longest space missions in history. The Landsat 5 Earth observation satellite ran from 1984 to 2013. And, of course, the Voyager 1 and Voyager 2 probes are still sending us data 40 years after their launches. And that's the news of the moment. This week, we dive into the world of CRISPR gene editing. Over the past few years, this gene editing technique has gotten a lot of press for the things it can do and the things it might be able to do in the future. It is a complicated topic, and I'm going to do my best to give you a general outline without losing both of us in the details. Consider this a brief snapshot into an exciting new scientific tool, and one that may be of great use far into the future. There will be links in the show notes to articles and videos that will help with all the details. I, in particular, found the videos very helpful, seeing a visual explanation of the CRISPR-Cas9 and how it works. What is CRISPR? Specifically, we're talking about CRISPR-Cas9, although there are other versions of this technique that are not Cas9, and some recent news stories have covered those. For the purpose of this show, I am going to shorthand everything to CRISPR. CRISPR, C-R-I-S-P-R, stands for Clustered Regularly Interspaced Short Palindromic Repeats, and along with the Cas9 enzyme, is in short a way to very specifically target a segment of DNA for editing. One very simple way I've heard it described is as molecular scissors that can snip and replace parts of a genome. Doing this isn't totally new, and there are several other ways to edit or alter genes in an organism, but CRISPR has proven to be very fast, versatile, precise, and simple, at least as simple as this kind of thing can get, and thus following up all those traits, also less expensive. It was discovered in the 1980s by people studying bacteria and their unique defense systems against viruses. The CRISPR-Cas9 technique, as with some other gene editing techniques, can be used to add or remove genes from a sequence, as well as turn genes on and off, thus altering their expression. This turning on and off of genes does not alter the DNA directly, and for some people has less ethical issues than in wholesale changing of the genome. Many researchers hope to be able to treat disease with this tool, and some treatments have already been tried. Given how much of our lives are controlled in one way or another by our DNA, there are endless possibilities. Many diseases, like sickle cell anemia, hepatitis B, muscular dystrophy, some cancers, and many others, have genetic causes. Even conditions like high cholesterol may be treated this way. In addition to treatments for disease, there are other uses for the CRISPR system. There is work, for example, being done to make mosquitoes immune to malaria, thus unable to pass that malaria on to people. Researchers who need to do animal tests need animals that get the diseases they are researching, like rats that get diabetes. It can be tricky to breed for a trait like that, so CRISPR could offer a shortcut. Perhaps CRISPR could even be used to ensure the viability of small populations of endangered species who are at risk for some sort of genetic collapse because there aren't enough breeding pairs left. CRISPR still faces challenges, and there is a lot of room for further development. The molecules don't always go where they are supposed to go to edit the correct cells, so methods for delivery need to be improved. Another roadblock for this and other gene editing methods is whether you are allowed to alter somatic cells, that is, any cell in the body not involved in reproduction, or if you can edit germline cells, those cells that are involved in reproduction. For example, if you treat someone's sickle cell anemia via their somatic cells, they will not pass that along to their offspring. Their offspring would then have a chance for themselves to develop sickle cell anemia, even though their parent no longer has it. If you can treat that in the germline cells, the cure will be passed on. There is also some question about how long a treatment will last. If you cure someone of a problem, like muscular dystrophy, will it only last until many of their muscle cells have been replaced by the normal functions of the body, thus renewing the defective cells? Also, apparently, to hold on to the fix as long as possible, the Cas9 enzyme must remain in your cells, which may provoke an immune response over time. Another concern surrounding CRISPR is in its potential misuse. If there is no oversight, someone with malicious intent might make a superbug that kills many people, or maybe even try to make super soldiers a la Captain America. Is it right to allow people to manipulate their children's DNA for future benefits, making them taller, more athletic, smarter, if we figure out what genes do what? And if someday something like that becomes available and is accepted by the public, is it only for the rich, or will it be available for everyone? If diseases are treated by CRISPR-Cas9, will the treatments be available to everyone or only available to the wealthy? There are many organizations, scientific and otherwise, and governments considering these questions now, and I think over the long run we will mostly see benefits. But like any tool, science can be used for good or bad, and it is vital for an informed and engaged public to be part of the discussion. As for the far future that I like to think about, the biggest thing for me isn't CRISPR itself, but gene editing overall. And if CRISPR proves to be the best way to do it, so be it. I can imagine being able to fix any disease or defect that comes along, even ones we don't know about yet. Or perhaps it will be a way to deal with the specific effects of space travel and off-Earth living, even maybe altering our genes to work better in the environment that we end up living in, like an accelerated evolution. Or perhaps gene editing can ensure the viability of smaller populations on generation ships. There are a few sci-fi stories I know of where distant colonies are settled first by embryos or just stored DNA sequences raised by robots or other means. For example, the Songs of Distant Earth book by Arthur C. Clarke and the Interstellar movie come to mind. I'm sure gene editing will play a huge role in something like that. It would be much easier to send 10,000 frozen embryos to a distant planet than 10,000 adults. Fully controlling our own DNA code just may be a key component of humanity lasting far, far into the future. All right, that is your show for this week. Thank you so much for joining me. I think I didn't mention that this was episode number 42 for those of you keeping track. Yes, indeed, 42. The answer to life, the universe, and everything is contained within this show if only you know how to ask the question. Anyway, thank you, as always, and come check out the show online at panfuture.org. Email me at sean@panfuture.org. That is SEAN. You can find links to Facebook and Twitter and iTunes and Stitcher and YouTube and all the other places you can find the show online. If you've got time, please write a review on iTunes or Stitcher or wherever you listen to the show. Share it with your friends. That's a great way to help out the show here. If you also just want to throw money at me, I will not complain about that either. Again, go to the website panfuture.org, and there is a PayPal donate link there as well. Unfortunately, I am not a 503c charitable organization. Maybe someday I can be. So your donations are not tax deductible, but you will have my gratitude. And maybe as the show grows, I'll be able to come up with some other things. I know a lot of podcasts have subscription options, micropayments, or you subscribe and you get something for it. Hopefully, maybe someday I can figure out some things to give you guys for chipping in to support the show. If you've got ideas, what kinds of things you would like? Do you want coffee mugs? Do you want pens? Do you want t-shirts? Do you want magnets? Do you want I don't know, what else is there? Whatever things. Do you want the things that would go with the donation? Let me know. And I guess that's it. I will talk to you again in the future.